Let us now look at how soil system gets organic carbon from plants. There are two major routes. One is above ground plant litter and it leachates, that is dissolved organic carbon washed into the soil from plant material by infiltrating rainfall. So this is one method. Another one is below ground root litter and exudation. This is collectively known as rhizodeposition. So these are all the two ways by which the organic carbon is moving from the soil system actually gets the organic carbon from the plants. Actually, how plants transport carbon to soil. So that is the overall process given here. Rhizodeposition consists of a continuous flow of carbon containing compounds from root to soil. So sugar, amino acid, sugar, alcohol, organic acids, these are all the organic compounds which are moving. And in the case of organic polymers like lignin, cellulose and hemicellulose, they require depolymerization by extracellular enzymes before they can be taken into the microbial cell and metabolized. So these are polymers. So the polymers has to be broken down. Then only they can be consumed. The relative magnitude of the various inputs from above and below ground will depend upon the plant species and in soil under agriculture crop management. So these are all the various things which control how soil gets the organic carbon. Now let us look at the organic carbon decomposition, because organic carbon now enters the soil and we are going to see how it is being decomposed. The various reactions are given. All the 6 reactions are the sequential reactions which are occurring. First one is the aerobic respiration. So here the organic carbon reacts with oxygen to give carbon dioxide and water. The second one is the denitrification reaction. Here nitrate ions are involved. So we will actually see each reactions in elaboration. The third one is the dissimilarity manganese reduction reaction. Here manganese 4 species is getting converted to manganese 2 species. Next one is the dissimilarity ion reduction reaction. Here Fe3 is getting reduced to Fe2. Then sulfate reduction reaction. Here sulfate ion is getting converted to sulfide ion. And the last one is the methanogenesis reaction, which produces methane and carbon dioxide. So these are all the various reactions and we will be looking at each individual reaction separately. So the primary terminal electron acceptor for organic matter decomposition in an aerobic sediment are nitrate, manganese 4, Fe3, sulfate and carbon dioxide. These are all the electron acceptors. So here are the reactions which are happening in the deep pristine aquifers. The first one, oxygen gets reduced and it is getting converted to nitrate, and this is again reduced to form the Mn4 species. Then this again undergoes reduction where Fe3 plus reactions are involved and followed by sulfate reduction, and finally we end up with the methane 1. This is in the deep pristine aquifer reaction. So the basic sequence of reactions are very same like whatever we have mentioned. And in the shallow aquifers contaminated with organic carbon, what will happen is the organic carbon is a starting point, and what we are seeing is exactly the opposite set of reactions what we have seen here. The organic carbon is now getting converted to methane, then sulfur dioxide reduction takes place, and iron and dissimilarity reactions of manganese, then nitrate and oxygen is released. So this is how the overall process that is happening. The first reaction is the aerobic respiration. So in the aerobic respiration is a process where glucose in the presence of oxygen is completely oxidized or decomposed in cell to release carbon dioxide, water and energy. So this is a common reaction most of the people already had studied about, and this produces large amount of ATP. The reaction occurs in 2 stages. One is the glycolysis reaction which occurs in the cytoplasm. So here the glucose is converted into pyruvate under anaerobic conditions. The second one is the tricarboxylic acid cycle or TCA cycle or Gerb's cycle or the citric acid cycle, which occurs in mitochondria. So under aerobic conditions, so the first one is an anaerobic reaction, second one is an aerobic reaction. Here the pyruvate is converted into carbon dioxide and water followed by oxidative phosphorylation to generate large amount of ATP. The second reaction is the denitrification reaction. So here different N oxides are successfully reduced by different enzymes to N2O, nitrous oxide or nitrogen. The first one is nitrate is reduced by nitrate reductase. So here nitrate reacts with 2 protons and 2 electrons to give nitrate ion and water molecule. The nitrate ion then reacts with another 2 hydrogen ions and 1 electron to give nitric oxide. This is basically a radical, and this releases water molecule. Then 2 nitric oxide reacts with 2 protons and 2 electrons to give nitrous oxide and water. The nitrous oxide then reacts with 2 hydrogen ions and 2 electrons to give nitrogen gas and water. So these are all the various reactions that are occurring. The overall reduction reaction can be shown as nitrate anion getting reacted with 10 electrons and 12 hydrogen atoms, protons to give nitrogen and 6 water molecule. The denitrifying microbes require a very low oxygen concentration of less than 10% as well as organic carbon for energy or for electron donor or as an electron donor. So here is the reaction that is given. So this is the organic source, the organic carbon that is involved. So 2 nitrate ion reacts with sugars to give nitrate and carbon dioxide and water. The nitrate ion further reacts with sugar molecules and the hydrogen atom to give nitrogen gas and carbon dioxide. So the third one is a dissimilarity manganese reduction reaction. Iron 3 and manganese 4 is an important mechanism for the oxidation of natural or contaminant organic compounds in diverse aquatic environments. So the dissimilarity iron or manganese reduction can be defined as the use of Fe3 or Mn4 respectively as an external electron acceptor in the metabolism. So iron 3 is reduced to iron 2, manganese 4 is reduced to Mn2, but in some cases what happens is Mn3 is also may be formed as an intermediate. And in the assimilarity, Fe3 or manganese 4 reduction, Fe3 or manganese 4 is reduced as part of the process of iron or manganese assimilation, that means being consumed by enzymes, cofactors and magnetosomes. The next one is the dissimilarity of manganese reduction. So here, under normal physiological conditions, the significant quantities of iron 2 and manganese 2 are only expected to accumulate outside the cell during dissimilarity iron 3 and manganese 4 reduction reactions. So the different types of dissimilarity reduces include fermentative Fe3 plus Fe3 and Mn4 reducers, that is glucose, or sulfur oxidizing Fe3 and Mn4 reducers, hydrogen oxidizing Fe3 and Mn4 reducers, organic acid oxidizing Fe3 and Mn4 reducers, especially formate, acetate, lactate and pyruvate, and aromatic compound oxidizing Fe3 reducers. So that means there are various sources are involved in this. Either it is a fermentative reaction or a sulfur involved reaction or hydrogen involved reaction or organic acid aromatic compound involved reactions. The fourth one is the dissimilarity ion reduction reaction. So here the reduction of Fe3 has been termed as the most important chemical change that takes place in the development of aerobic soils and sediments. So here is the complex organic matter, and we also have iron 3 plus exist in that soil. So what happens, one of the reaction is a hydrolysis. So there are different types of hydrolysis reactions are occurring. One is the hydrolysis which leads to aromatics, and this aromatics is further getting converted to carbon dioxide and Fe2 plus, or hydrolysis of long chain fatty acids are happening which leads to propionate and acetate and butyrate, which further getting converted to carbon dioxide and iron 2. Or sugars and amino acids are getting converted to hydrogen and formate, which further getting converted to carbon dioxide. So these are all the various cycles that are involved. Then the next one is the sulfate reduction reaction. In the sulfate reduction is a redox reaction in which a divalent anion sulfate, so for 2 minus, is reduced to sulfide is 2 minus, and it involves addition of 8 electrons. See, during this assimilation, the sulfide is incorporated into sulfur containing amino acids, the mainly methionine, cysteine, homocysteine and taurine. During the dissimilarity sulfate reduction, the sulfide is converted to hydrogen sulfide and thiols or S2- sulfides and other aqueous species depending on the solution pH and composition. These reactions can be coupled to the oxidation of other chemical species including a variety of organic compounds like acetate, pyruvate and methane. It is important diagnostic process that oxidizes organic carbon generated by photosynthesis. So this is the most crucial part of sulfate reduction. The dissimilarity sulfate reduction is a major driver of the global carbon cycle. So the various reactions are given here. Hydrogen reacts with the sulfate and anion in the presence of H plus gives 4 water and thiol. Thiolate anion SH minus. 2-formate reacts with sulfate in the presence of H plus gives bicarbonate and thiolate anion. Acetate also produces thiolate and bicarbonates. This is pyruvate. And we also have propionate and we have methane. So these are all the various things we have seen and all of them undergo this dissimilarity sulfate reduction to produce in most of the cases thiolate anions. The last one is the methanogenesis reaction. So in the methanogenesis is a process where reduction of carbon dioxide, formate or carbon monoxide, that is basically the carbon dioxide reducing methanogens, methanol or methylamines or acetate produce methane. So the final product is methane. So that's the reason the reaction is called methanogenesis, that is formation of methane from various different sources. Here are some of the reactions given. We will be studying one entire module about methanogenesis reaction. So there we will elaborately study each individual steps. The first one is 4 hydrogen reacting with the carbon dioxide to give methane and water. This is carbon dioxide reducing methanogens follow this particular process. Methanol is getting converted to methane, carbon dioxide and water. This is carried out by methylotropic methanogens. Formic acid, acetic acid getting converted to methane and carbon dioxide is carried out by acetoclastic methanogens.